Hello everyone, welcome to Supplement Tutorials. Today we'll be talking about anemia of lead poisoning. And before we begin, I would like you to hit on the subscribe button to subscribe to this channel. Alright, now in our previous anemia lectures, we talked about the microcytic hypochromic anemias. So we talked about iron deficiency anemia, and then we talked about the thalassemias. So today, our next anemia we'll be discussing, which is a microcytic hypochromic anemia, is the anemia of lead poisoning. So in our previous lecture, we said we have anemia can be categorized. So we have the microcytic anemias. Then we have the macro or the megaloblastic. So under the microcytic anemias, we talked about iron deficiency anemia, iron deficiency. We talked about the thalassemias. Then we have the lead poisoning, anemia of lead poisoning. Then under the macrocytic, we have the vitamin B12 deficiency anemia. Then we have alcohol related anemia. Alright, so these are the two types of anemia we talked about. We are to talk about here. So we've already talked about the microcytic anemias, especially the iron deficiency anemia and the thalassemia. So today we are going to talk about the lead poisoning or the anemia of lead poisoning. In future videos we'll talk about the vitamin B12 deficiency and then the alcohol related and other anemias, especially the anemias that result in hemoglobinopathies, or that is related with hemoglobinopathies, that's the sickle cell anemias and etc. Alright, so today we are going to talk about the anemia of lead poisoning, and we are going to discuss the causes, the pathophysiology, and then the medical diagnosis and the symptoms as well of anemia of lead poisoning. So first of all, what we have to know is that anemia of lead poisoning is a microcytic hypochromic anemia. It is mostly microcytic hypochromic anemia, and it is rarely a normocytic anemia. When we say a microcytic hypochromic, it means they have a small cell size and then a larger central pallor or a pale color. That is why we call it what? A hypochromic or a microcytic hypochromic. So the MCV, which represents the size of the red cells, is small, and then the MCH, that's the mean cell hemoglobin, is also what? Is also small. So this will tell you that it's a what? It's a microcytic hypochromic anemia. Now let's talk about the causes of lead poisoning, anemia of lead poisoning. So the causes, we have acute or chronic exposure to lead. That is basically the main cause of lead poisoning or anemia of lead poisoning. Acute or chronic exposure to lead. Now we have the sources of exposure. We have environmental sources, we have occupational sources, then we have ingestion. So in the environmental sources, we have most of the lead compounds in our environment. We have most of the lead compounds in our environment. We have some in the soil, so contaminated soil and water. Then we have lead based paints. So interaction with lead based paints, you can also be exposed to lead poisoning. Then the occupational exposure, so people who are mostly found or who are mostly miners. So the miners basically use heavy metals in their mining. So they mostly use mercury, and what and lead, and other occupation that makes, so some paint industries, they also make use of what, of the lead. Then ingestion, so this mostly happens in children and some adults. So when you have um, lead pipes, they can cause lead poisoning, so they can basically leak into the water and then cause lead poisoning. So lead pipes, through drinking water from lead pipes. So lead pipes, so when you drink water from lead pipes, you can get what, lead into your system. Then you can talk about paint chips. So there are some children, they might mistakenly consume paint chips, and then they will find themselves having lead in their system. So let's talk about how lead poisoning causes anemia. That is the pathophysiology of lead poisoning. So first of all, we'll talk about hemoglobin synthesis disruption. Hemoglobin synthesis disruption. Then impaired RBC survival through oxidative burst and RNA degeneration. They will talk about bone marrow toxicity. They will talk about competition with other metals. So pretty quick, let's talk about the hemoglobin synthesis disruption. So before we talk about the hemoglobin synthesis disruption, we have to first know what hemoglobin synthesis, or how hemoglobin synthesis occurs. So we have lectures on hemoglobin synthesis. You can go and check that one out. So here in hemoglobin synthesis, we have succinyl CoA reacting with glycine to give us what, alpha aminolevulinic acid. So this alpha aminolevulinic acid leaves the cells, and then they form what, porphobilinogen. Then they form uroporphrinogen 3. Then they form coproporphyrin. Coproporphrinogen 3. Then it moves back into the cell. Then coproporphrinogen 3 forms protoporphyrin IXA. Then protoporphrinogen IX. Then protoporphyrin. Then hem. So that is basically the hemoglobin synthesis, or the hem synthesis. So this hem will get fused with iron, and then it will form the heme. Then it will combine with amino acids, basically the globulins protein, to form the hemoglobin. So that is what hemoglobin is about. So this is what a brief description of hemoglobin synthesis. Succinyl CoA combining with glycine to give you what, alpha aminolevulinic acid. Then this alpha aminolevulinic acid will leave the cell, and then outside the cell it will be converted to what, to porphobilinogen. Then from porphobilinogen to form uroporphrinogen. The uroporphrinogen will form a coproporphrinogen 3. Then it will move back into the cell. Then from coproporphrinogen 3 to form protoporphrinogen IX. Then protoporphrinogen IX will form protoporphyrin IX. Then it will form what, heme. Okay, so iron is going to fuse. That is iron 2 plus is going to fuse with the protoporphyrin IXA to form what, the hem. And the hem will combine with globulin proteins to form the hemoglobin. So this is basically what hemoglobin synthesis is. Now, what lead poisoning does is that it interrupts the conversion of alpha aminolevulinic acid into what, into porphobilinogen. So this is what happened. So how lead disrupts hemoglobin synthesis? So lead interferes with the conversion of alpha aminolevulinic acid into what, porphobilinogen, leading to the accumulation of what, of alpha, of delta, sorry, this one is delta aminolevulinic acid in the urine and then in the blood. So that is the first thing it does. So first of all, it inhibits the what? The conversion of ALA into what? Or into porphobilinogen. Now when this happens, it cuts the process. The process is to continue from the side. So the process end at this side. There is no conversion into porphobilinogen, and all this process will not be able to be possible. Therefore, there is not going to be any hem synthesis, and therefore no hemoglobin synthesis. And this, as a result, will lead to what? Less hemoglobin formation, and as a result, will lead to anemia. So that also accounts for its what? Hypochromicity. Then the second thing it does is that it prevents the incorporation of iron into protoporphyrin IXA to form heme. Now, this is what happened, as I said earlier. So, protoporphyrin IXA combines with iron, Fe2 plus, then it forms the heme. Now, here lies the case: the lead is also a metal, and then the iron 2 plus is also a metal. So, there's actually some form of competition. Between the lead and then the iron. And what happens is that this competition most at times prevents the incorporation of iron into the protoporphyrin IXA moiety to form the heme. So that is also one reason why lead can lead to anemia. Okay, now let's talk about the impaired survival of RBC. So lead in its nature is a strong inducer of reactive oxidant species, and also a strong neutralizer of antioxidants in the body. So the body actually is most at times exposed to reactive oxidant species or oxidative species. And these reactive oxidant species, also known as ROS, induces oxidative effect on cells. And one of the ways our cells prevent itself from this oxidative burst is through antioxidant means. So we have natural antioxidant means in the body. We have superoxide dismutase. So we also have the glutathione. There are a lot we have. So what happens is that this lead neutralizes these natural antioxidants that we have in the body. And once these ones are neutralized, it can no more fight against the oxidative stress that is being imposed on the cells. So the cells will eventually undergo oxidative burst. And once the cell undergoes oxidative burst, it means the membrane of the cell disrupts, and then whatever content that the cell has is exposed. So in the case of red blood cells, all the hemoglobins are going to be exposed, and they are going to move out, or they are going to diffuse somewhere into the interstitial space or somewhere in the vascular compartment. And as a result, the more the red cells are breaking down, the more person experiences anemia. So that is it. So they also enhance the activity of reactive oxidant species. So there are also other reactive oxidant species that we have in our body. And what this lead does is that they increase their activities. So once the activity increases, it increases the oxidative stress on the red cells, and therefore it increases oxidative burst. So that is also another way the lead causes anemia. And most of the times the lead also causes what we call RNA. It disrupts RNA degradation. So in the process of hematopoiesis, what happens is that the nucleus eventually disappears from the red cells. That's why we know red cells are enucleated. They don't have any nucleus. So when it reaches some stage, like the reticulocyte stage or the stage before the reticulocyte stage, the RNA fragment begins to disappear from the red cells. Now, what lead does is that it prevents this RNA degradation, and therefore it is one diagnostic marker to tell if someone has what anemia of lead poisoning, so you see what you see basophilic stipplings of what RNA remnants in the cells, in the red cells. And once you see those ones, you should know that this plus other diagnostic markers, you should know that the person's condition is gearing towards anemia of lead poisoning. Alright, so that should also be in your head. And now we have bone marrow toxicity. So this basically inhibits what? Inhibits bone marrow function. So they inhibit erythropoietic stem cells from undergoing what hematopoiesis or erythropoiesis. So the lead also directly affects the bone marrow, and then they inhibit what hematopoiesis. Now let's look at the symptoms. So we have anemia related symptoms. So this one, the general symptoms of anemia, we can talk about pallor in what? In conjunctiva. We can talk about what? We can talk about tiredness or fatigue. So we can talk about okay, so tiredness, fatigue. So all the general symptoms of anemia that we can possibly talk about. So rapid breathing, so frequent tiredness, rapid breathing, pallor in conjunctiva, etc., etc. So all these symptoms of anemia, dizziness, etc., tells us what the anemia related symptoms of anemia of lead poisoning. And lead poisoning generally can also cause what? Lead toxicity. So this lead toxicity is an extreme form of what? Of lead poisoning. So in this case, it can cause what? Cognitive dysfunction. So it can cause cognitive dysfunctioning, etc., etc. So mostly the person begins to have some neurological dysfunction and sometimes to renal dysfunction. Renal dysfunction. Alright. So here are some laboratory diagnoses that we can use to tell if someone really has anemia of lead poisoning. So we have CBC. So CBC, what we are looking for is the hypochromicity and the micro is the hypochromicity and how microcytic the cells are. So we are looking forward to seeing low MCVs, low MCH. We are looking forward to see low hemoglobin 2 as well. We are looking forward to see low RBCs as well. Then we are also looking forward to see anisocytosis and then poikilocytosis. So when we say anisocytosis, we are talking about variation in red cell hot sizes. So this is variation in red cell sizes. Then here we can talk about what? Poikilocytosis. We can talk about variation in RBC hot shape. Then for peripheral blood lead levels, to that one is a direct indicator. When it's so much, we can say that it's what lead poisoning. Blood smear, we are looking for what? Basophilic stipplings, which is also a diagnostic matter. And then for the zinc protoporphyrin, now remember that I said one of the function or one of the disruption of the lead to the hemoglobin synthesis is the prevention of what? Of protoporphyrin ISA from what? From uptaking iron. So mostly if that does not happen, It is zinc that binds the what to the protoporphyrin to form a substance we call zinc photoporphyrin. And this substance does not produce what? Hemoglobin or hem, because the only thing that we know that is capable of combining with protoporphyrin in order to form hem is what? Is iron. So when you find this, it means there has been an error somewhere, and it's also an indicative of what? Lead poisoning or anemia of lead poisoning. Alright, so thank you very much for watching this video. In our next video, we'll go forward to discussing the other types of anemia, such as the megaloblastic anemia. See you next time. Before you exit, I would like you to hit on the subscribe button to subscribe to this channel. Thank you very much.